Hello, my name is Jody Schwartz and I'm an associate professor at Vassar College, and I'm really excited to be able to talk with you today a little bit about the research that I do on corals. And so I will be talking today about coral reefs in a warming world. Before I get into the topic of coral reefs, I just want to explain a little bit about who I am. As I mentioned, I'm in the biology department where I am very lucky to be able to teach in my area of expertise of genetics and genomics and bioinformatics. And even more than that, to be able to bring into the classroom some of my research interests in the area of corals. Here I am working on a project on coral bleaching in the Caribbean. And also in the Bermuda Institute of Ocean Sciences, where I was lucky to be able to put together my research and teaching interests to teach an environmental studies in the field course a few years ago, where students traveled with me to the Bermuda Institute of Ocean Sciences to develop a variety of independent projects on corals and coral reefs. So you may have heard and probably have heard that corals are often considered the rainforest of the oceans, and that's because they serve as the foundation of the ecosystem. Corals are animals. They're animals that produce skeleton, and much like trees, they create the three-dimensional structure and also the food, the base of the food chain for the entire ecosystem that then consists of thousands of species of invertebrates and at least hundreds of species of fish, as well as a myriad of microscopic organisms. Both algae and bacteria. Coral reefs are incredibly significant economically and in their ecosystem services. They protect coastlines from erosion. They serve as nurseries for fish that develop billions of dollars of fisheries. They're important for tourism and recreation. And although they only comprise about 1% of the ocean floor, they comprise about 25% of all marine life. So they are quite significant. They also are one of the most endangered ecosystems on the planet. And as this New York Times article is showing, 2019 was a record year for ocean temperatures. And as ocean temperatures rise, this increase in ocean temperature is incredibly detrimental to corals. Corals form a symbiosis with microscopic algae that live inside the cells of the coral. But when corals are exposed to a change in seawater temperature of just 1 or 2 degrees, this causes a catastrophic destruction to the symbiotic relationship between the coral and the alga. And the result is that the algae, which are golden brown color, are jettisoned from the coral, leaving the coral a stark white color. This phenomenon called coral bleaching is not new. This is not something that has just started occurring in the past few years. The first coral bleaching events, though, were local events. The first ones were observed in the 1980s, and they would occur in small patches of individual reefs. But starting in 1998, the first global coral bleaching event occurred, where corals bleached across multiple regions across a broad geographic area. And ever since then, coral bleaching events have become more and more common and more expansive in their extent. Corals really are the proverbial canary in the coal mine. They've been one of the first ecosystems documented to respond to climate change, and they are one of the first ecosystems that are possibly endangered or almost going extinct in some areas. So coral reefs provide critically important ecosystem and economic services, and they are highly impacted by warming ocean temperatures. And that is at the heart of the research that I do, is understanding the symbiotic relationship of corals with their microalgae in order to understand why corals bleach, how they bleach, and how we can ameliorate or mitigate the bleaching response. One of the things that I find most fascinating about corals is that as animals, they also are geologic structures. They produce a calcium carbonate skeleton that ultimately forms the rock of the reef ecosystem. Shown here is a colony, Acropora, in the Caribbean, so this is in the United States. This Acropora colony is made up of thousands of small polyps. So while this entire colony is about 5 feet across, the individual polyps are about a millimeter across. And so zoomed in here on a collection of individual polyps, you can see each polyp has a mouth and is ringed by tentacles. These tentacles capture food from the water and pull it into the mouth where it's digested. These coral polyps also are each individually secreting a calcium carbonate skeleton that it lives on top of. Okay, so think of it as the coral making a little chalk cup that it sits inside of. And over time, these individual cups build up to create layers of skeletal material. And shown here in the bottom right is a coral skeleton with no polyps living on it. It's just the bare skeleton. And you can see the remnants, the cups, from the individual polyps that once lived on top of this skeleton. So the fact that corals are animals that produce rock, I find utterly fascinating. And the fact that that rock then builds up to create these underwater environments, to me, is even more fascinating. And so one of the things that captured my interest about corals early on is the fact that these biodiverse ecosystems occur in regions of the ocean that are very nutrient poor. For a food web to begin, there has to be the presence of nutrients to support plant growth. Without plant growth, you can't have animal growth, and you can't have an ecosystem. So corals tend to grow in regions of the ocean like the Sargasso Sea area that are very nutrient poor and therefore have very little plant growth. Contrast this to the Monterey Bay, which is very nutrient rich, and you can see this scuba diver is swimming in almost an emerald green water. That emerald green is imparted by the billions of microscopic algae that are living in the water, and also you can see a little bit of kelp, the seaweeds that grow. The algae and the kelp form the basis of the ecosystem in the Monterey Bay because there is plenty of nutrients for those algae and kelp to grow. But in tropical regions of the ocean, these are nutrient poor, and so the marine ecosystem has very little in the way of nutrients to get it kick-started. And this is really what captured my interest in studying corals. Corals are able to grow in these nutrient-poor environments because they have formed a nutrient recycling symbiosis with algae. Corals recycle nutrients through the symbiotic relationship with microscopic algae that live inside their cells. The algae are photosynthetic and produce sugars. They transfer this sugar to the coral, where the coral burns it for its metabolic needs, and in return, the coral growth produces waste products that are rich in nutrients like nitrogen, and this serves as fertilizer to support the growth of the algae living inside of them. So corals manage to capture what little nutrients are in the water and recycle it back and forth between a primary producer, the algae, and a consumer, the coral. And together, this forms the basis of the ecosystem for other things to live in and on and to have a source for food. So just to give you a sense of what this looks like, here's an individual coral polyp that has no symbiotic algae in it, and you can see that it's a stark white color. This coral polyp as an animal is producing nitrogen waste, and that nitrogen waste is used by the algae to fuel their own growth. And in return, the algae are performing photosynthesis and providing sugar to the coral polyp. Together, these two partners form this beautiful golden brown polyp that is sometimes called the holobiont, as it represents both animal and algae. When corals bleach, if I were to expose this polyp to an increase in temperature of 1 or 2 degrees, the polyp would expel the algae living inside their cells and render it back to this non-symbiotic condition that is stark white. And so you can see why coral bleaching is called coral bleaching. It's because they are rendered from a golden brown color to a white color due to the loss of their symbiotic algae. To give you a sense of the coral bleaching event, here is a region of reef from 2014 looking like its beautiful golden brown color until it experiences an increase in water temperature of a couple of degrees and the coral bleaches. This, of course, is going to be very detrimental to the ecosystem because healthy coral reefs contain dense stands of coral and support high levels of biodiversity because of the productivity of the coral symbiosis. Once coral bleaching occurs and the corals are rendered non-symbiotic, they no longer have access to all that metabolic carbon that they had been acquiring from their symbiotic partners. And this coral bleaching then can, if it's protracted, can kill the corals. And if that is followed by some kind of massive storm or hurricane, the coral reef ecosystem begins to decay. Corals cannot rebuild, they can't grow, they cannot reproduce, they cannot seed new regions of the reef. So coral bleaching has a domino effect of leading to the degradation of the entire reef ecosystem. So what can be done about coral bleaching? Well, one of the things that we can do at Vassar is learn about corals. Even though we are in Poughkeepsie, there are many things that we can do to learn about corals. One of the things that we can do is offer courses that allow students to study corals either in the lab or in the field, as we did in the environmental studies course. Another thing that we can do at Vassar is to do basic science to understand corals. We can understand or learn about the coral symbiosis and how that is affected by climate change. We also can understand questions about coral reproduction and how can we regenerate corals after bleaching. Another thing that we can do is work to restore degraded reefs, and there are a number of global efforts that are working to farm or garden corals for replacement on the reef. And of course, the biggest thing that we can do, need to do, and is one of the hardest things to do, is to globally reduce carbon emissions and other greenhouse gases so as to slow or hopefully reverse the impacts of rising temperatures. So this is a slide that's showing you a collection of different projects that my students have worked on, and I'm not going to focus on these particular projects. I'm going to tell you a little bit more today about our efforts to understand coral reproduction. This slide is showing us the primary work that I have focused on until recently, which is to dissect the cellular and molecular mechanisms that lead to bleaching. And we do that through a variety of different genetic and genomic approaches. Other work involves identifying genetic strains of symbionts that may help be more resilient to increasing water temperatures. I've had a couple of students work on questions about how to mitigate the bleaching response, for example, exposing corals to antioxidant compounds that help reduce oxidative stress. Oxidative stress is thought to be the primary biological process that causes bleaching. As the coral cells experience oxidative stress, that causes cells to either decay, be removed from the coral, or otherwise expel the symbionts. All of these things lead to bleaching. Today, what I'd like to spend the rest of my time on is to tell you a little bit about the work that my lab has done over the past few years, thinking about how coral reproduction is affected by warming oceans, but also how we might use coral reproduction to think about ways to restore, conserve, and protect reefs. This slide is showing one of my collaborators' labs in Taiwan, which is at the National Marine Biology and Marine Science Aquarium in Taiwan, where a number of tanks have been set up in actually a really vast research area that pipes in water from the marine environment so that researchers can grow corals and study all sorts of different questions related to coral symbiosis and coral reproduction. In order to think about coral reproduction, it's really helpful to look at the life cycle of a coral. So I'm going to just lead you through how is a coral born and how does it reproduce. In the lower left here, we can see an example of a mature coral colony like the one I showed earlier. This coral colony is made up of many individual polyps. Each one of these polyps produces either eggs or sperm or both. Some corals are hermaphroditic, and they release these eggs and sperm into the water, where fertilization occurs in the water. Fertilization of the eggs occurs in the water, and those eggs go on to develop a coral embryo through cell division, which develops into a little swimming larva that looks like a little barrel with little cilia on it. That embryo or that larva swims around until it finds a place that it wants to settle onto the ocean floor, and it undergoes a process called settlement, where it settles to the ocean floor and undergoes metamorphosis into a polyp. So it develops tentacles, and now this is a brand new coral colony waiting to develop from an individual polyp that will then undergo asexual reproduction to form more genetically identical polyps to produce the colony. One of the most fascinating things about coral reproduction is that it occurs in a highly synchronous and timed way. Corals produce in these exquisitely timed mass spawning events where an individual species will release eggs and sperm at the same time over just a period of hours. Shown here in this nature video, you can see an individual coral colony. If we zoom in on that coral colony, we can see the individual polyps that make up that colony, and each one of the polyps has this little pink round egg sperm bundle just poised in their mouth, ready to release. At some moment, and in response to some cue that's yet to be understood, the corals will release their egg sperm bundles all at once. So this colony, the hundreds or thousands of polyps that make up this colony, release them all at the same time, and the egg sperm bundles rise up through the water and collect at the ocean surface, where the eggs will be fertilized, and that life cycle that I showed to you on a previous slide will commence. But amazingly, the coral colonies across the reef, all from the same species, will all be undergoing this timed release at the same time. And I'm just fascinated by trying to understand what are the biological or hormonal cues that cause that to occur. Some of the questions that I'm most interested in addressing have to do with the underlying biological processes that lead to the coordination of the spawning response. And we know already that the timing of spawning is coordinated through environmental cues, such as the changing seasonal seawater temperatures, monthly moonlight cycles, the pattern of daylight and dark through the daily cycle, and also possibly through the cues of hormones that might be released by corals that help other corals determine when to spawn. Because corals only spawn once or twice a year, it is very challenging to do any research on coral spawning and to understand what controls their reproduction, because they only spawn a couple times a year. And so one of my interests has been in finding an experimentally tractable model system to examine the timing of spawning. And I'm going to talk to you about one of them that we've developed. This is a sea anemone called Nematostella vectensis. The image that I'm showing on the left is an individual Nematostella polyp that's probably a couple centimeters long, so they're quite small. And we use this Nematostella in the lab to understand its pattern of egg release. This video I'm showing here was taken in my lab. It's showing a Nematostella polyp with eggs filling its pharynx, or the area right below its mouth, and then release of the eggs into the water. Okay, so on a camera we can capture this moment of spawning when eggs are being released into the dish. And our underlying question, our very first question, is what is the baseline timing of spawning? When Nematostella are exposed to the environmental cues that cause its spawning, how much time elapses before they start spawning? Does the spawning occur over a period of minutes or hours? These are some basic questions that have yet to be answered. So in some preliminary experiments this summer with URSI students, two students, Chloe and Sedona, and I examined this question about the baseline timing of spawning. We exposed the Nematostella to environmental cues of elevated temperature and light, and we used a camera system to photo document their spawning behavior every 5 minutes over 10 hours. The result is that we got a series of photos that Sedona and Chloe looked through to identify some key stages of spawning. And here you can see an image on the left at 520 minutes after the initial environmental cues began with a polyp. And you can see at 525 minutes, you can see these little white dots that are the eggs filling the pharynx. And at 535 minutes, you can see that the eggs have been deposited into the dish. So we can precisely time when each individual polyp in this dish of polyps is spawning. The result of that is that Sedona and Chloe collected a lot of data this summer and analyzed this data, showing that over a series of different spawning trials, we did approximately 8 or 10 trials, that the anemones tend to spawn between about 500 and 600 minutes after the initial exposure to the environmental cues. This is really helpful information because now we can do experiments where we expose anemones to different environmental conditions, including increased water temperature, and examine whether that causes shifts in the timing of their reproduction. This kind of information can then be used to help us understand how corals might be responding to climate change, but also how can we help corals successfully reproduce so that new reefs can be formed and so that reefs can be restored. So that's just a tiny snippet of the research that I do, but I hope that you got a sense that at Vassar we really do blend together what happens in the classroom with what's happening in our research lab and allow students to develop projects that are of interest to them, but that also help the faculty with their research. So coral reefs in a warming world. We've talked about some of the challenges that coral reefs face and also potentially some of the ways that basic science and conservation science can help corals. So my question that I would like to pose is, will humans become the symbiotic partner of corals to help protect, conserve, and evolve solutions to coral bleaching? And I hope that my students and I are helping to do that we are forming a symbiosis with corals. And finally, I would like to acknowledge that symbiosis research itself is symbiotic. I've worked with so many different people over the years, some amazing students. Vassar students are just amazing, and they go on to do amazing things. Shown on the left here are a collection of students over the years who have worked on various aspects of either the reproduction project or the coral symbiosis and bleaching projects. And on the right are collaborators at other institutions that I have worked with. And finally, the funding sources that have helped support not only my research, but my teaching and mentoring activities with Vassar students. I really appreciate this opportunity to share with you a little bit about what I do, and I would be open to emails or phone calls if you're interested in learning more. Thank you very much.